a petals, it needs a combination between B and C. Need to be come together, right? Working together, and then it can form A and B. It can form petals. While stamens needs a genes from B and C. All right. So carpels it needs C alone. All right. So depending on what wheels that we are, what floral organs that we are talking, it depends on the system here, like A, B, and C. All right. A and C are equally dominant. B engines are always always expressed in associate with A and C. Expression A gene alone will form wheel that is separate. All right. So kalau okay. So kalau A only, dia akan produce sepal. Alright. So kalau B, A and B and sepal. Okay, so look here. Leceh. Okay. So kita dah tahu what is A, B and C. Alright. What genes that need to form any set of the floral organs. Okay. Let's say kita nak tengok actually macam mana actually kita tahu yang A alone ni produce sepal. Alright. Macam mana kita boleh tahu that petal needs A and B. Alright. Macam mana kita tahu yang petals ni bukan dikontrol oleh A only. It needs to be A and B. Right? We need to do a mutation. Mutation ni kita kena prevent any transcription ataupun translation of A genes to be translated into proteins. Okay. You know during I assume that you know during the gene expression class, right? The transcription, right, will produce mRNA, and the mRNA will enter the ribosome complex and it will be translated into proteins, right? Whether the mutation can be done either you disrupt ataupun you interrupt the sequence in the nucleus, right? The sequence of the genes in the nucleus so that the transcription, okay, sepatutnya contoh resepi nak buat nasi lemak, alright, resepi nak buat nasi lemak ada dia punya resepi lah, dia ingredient bla 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 bla, alright. That recipe of nasi lemak is the DNA, alright, the DNA sequence untuk produce nasi lemak, alright. So bila produce nasi lemak, apabila tukang masak tengok resepi resepi tu adalah DNA sequence, resepi nasi lemak, right? So tukang masak akan baca resepi nasi lemak, alright, dan translated into a physical nasi lemak dan hasilkan nasi lemak. So the tukang masak tu adalah mRNA, alright. So tukang masak akan copy resepi nasi lemak, dia adalah mRNA, messenger mRNA. So tukang nasi lemak ni tadi dia akan tukar mRNA kepada protein, which is nasi lemak lah, right? Nasi lemak analogically. So what you do, you can disrupt the recipe of the nasi lemak in the book, right? So that the tukang masak mRNA tadi, tukang masak tadi tu dia akan copy a wrong recipe of nasi lemak, right? So bila translated, bila dimasak, ia tidak akan menjadikan dia tidak akan menjadikan it will not be translated into nasi lemak. Different things lah, whether nasi dagang ke nasi goreng kampung ke nasi goreng cili. So ataupun dia memang tak dihasilkan langsung, right? So first you can disrupt the gene DNA sequence, right, in the nucleus, ataupun resepi betul, right? Resepi betul, and then tukang masak, dia copy resepi yang betul, right? Copy resepi tu, just imagine it was an mRNA. Dia dah copy resepi yang betul of nasi lemak, right? On the way dia nak pergi cooking untuk menghasilkan the right nasi lemak, somebody else knock the chef out, mati, kill the chef. Kill the chef. So nasi lemak takkan dapat dihasilkan. So the objective of this mutation is to look at the function of the proteins. Okay, it's very difficult to remove the proteins. Okay, protein will always be, right? You kena buang from the akar. You need to tackle from the akar. Okay, you cannot tackle from the protein level because proteins will always be produced as long selagi mana the mRNA tu are being produced, transcription happen. As long as the transcription happens, so the proteins will always be produced. So what you need to do, instead of removing out the proteins, you need to disrupt the change of the transcription. Alright, whether you want to kacau the mRNA ataupun you kacau the DNA sequence, right? So bila protein tak dapat dihasilkan for example in this case is gene A, you kacau the recipe of the gene A ataupun do you kacau the mRNA of the transcription in the gene A, right? So the proteins for the gene A ni takkan dapat dihasilkan, alright? Takkan dapat dihasilkan, so kita akan panggil mutant, alright? So daripada situlah kita boleh tahu, okay, kalau we knock out the gene A, protein A will not be produced, so it will not produce a sepal. Alright? And also petals, because petal needs A and B. Alright? So kalau you mutate gene A, alright, you will not see petal and sepal. So what you will see adalah stamens and carpel. So like here, stamen and carpel. Okay. Oh, sorry. Okay, see the stamen is the blue one, carpel is in the brown one, and the petal is the yellow one, the sepal is the green one. So if you remove the gene A, right, mutate the gene A, you akan menghasilkan carpel and then stamen saja. Alright, B and C, and then C. Alright, so when gene A is mutated, normal functional, C gene will become more active because it will change the pill formation. So mutation A gene for Arabidopsis is called apetala2. Right? Kita panggil mutation tu memang apetala2. Maknanya ketiadaan proteins apetala2. Dia namakan apetala2. Right? So if you do a mutation of the B genes, okay, the concept is same. How are you going to be doing on the mutation of the B genes? The same way that you change or you disrupt or you kacau anything lah in the gene sequence, alright? Ataupun you change on how the mRNA ataupun the transcription, right? Macam tukar masak, buku resipi nasi lemak, tukar masak, and then the product, so that will be the same lah. Alright. So what you will see adalah sepals and carpel, right? Dua benda. Alright. So the gene mutation of the B genes kita panggil apetala3 ataupun pistillata apetala3. Biasanya apetala3. Alright. So if you mutate the C genes, what you will see adalah so dia akan menghasilkan sepal and petals lah. right? So dia akan menghasilkan dua tu saja, right? So this is the summary. Wild type is pokok yang asal-asal wild types. Okay, so if you mutate the gene A, what you will see couples and what the orange ones? Or stamen sorry, if you mutate the gene A, you will see stamens and carpels. When you mutate genes B, you will see stamens and sepal, right? And mutate C genes, you will see petals and sepals only, right? Okay, enough with the gene expression or factors that controlling uh, organs in the flower during the flower developments. Okay, and then we go to the floral evocation. Okay, evocation. A floral evocation requires the apical buds. Okay, it's the process of the transition from the apical buds to the apa ni SAMs shoot apical meristems kepada floral apex, alright, from the shoot apex kepada floral apex. It requires two stages, alright, ataupun two developmental stages. One is competence, another one is developmental, alright. Sorry, one is competence, another one is determination, alright. So we look at the competence first. What is competence? A competent vegetative meristem is one that can respond to a floral stimulus by becoming florally determined. Okay, it's committed to producing a flowers. Okay, the competence plants atau buds able to respond in expected manner when given the appropriate developmental signals. Induction, okay, normally photoperiods. Okay, generally flowering is induced by a photoperiod. Okay, nanti kita akan belajar ada short day periods, a long day plant, short day plants. Okay, the amount of energy ataupun an amount of light, okay, ataupun the duration of the light, okay, is an induction factors ataupun stimulus, okay, whether plants can start flowering or not. Okay, it's just induction. Okay, the duration of the light is just induction, but at the end of the day, it still depends on the plant itself whether it has a competency to do transition from the shoot apical meristem kepada floral meristems. It doesn't mean that at the induction from the right photoperiods of light, they akan induce all the plants to be flowering. It's not like that. It still depends on the readiness of the plant itself. All right, and then determines able to follow the same developmental programs even after removal from its normal position in plants. Okay, we're going to discuss this a bit more details later, right? Given the signals, normally hormones, and then the plants can start flowering, all right? Okay, the competence ni adalah, for example, if a vegetative shoot season, saya tak ingat nak pronounce this. Cyan, alright. If a vegetative shoot science is grafted onto a flowering stalks and the cyan flowers immediately, it is responding Okay, it is says that there's the cyan to okay, so the stalk to dah dipanggil competent. Okay, for example, you ada pokok stalk. Okay, kita kalau belajar propagation, kita akan ada mata tunas, what we call scion, and also stalk, parent, pokok penanti. Alright, so nak tahu pokok penanti tu dia dah boleh flower or not, okay, dah pass through the competence stage or not, kita ambil mana-mana vegetative shoot, alright, pokok lain, alright, same species lah, and then you do a grafting, cantuman mata tunas. If the vegetative shoot to immediately flowerings, so that means the floral stimulus present in the stalk tu memang dah competence, right? Means dah ready to start flowering. Even though you masuk vegetative shoot pun dia akan start flowering. Alright. Failure of the scion to flower will indicate that the shoot apical meristem has not shoot apical meristem has not yet attained competence. And then the second is determination. Okay, how determines of the plants to go and entering the flowering stage. A bud is said to be determined if it progresses to the next developmental stages, which is flowering after being removed from its normal context. Thus, a florally determined bud will produce flowers even it is grafted onto a vegetative plant that is not producing any floral stimulus. So means that if pokok tu dah determines to flower, the pucuk, kalau dia dah determines to start flowering, even though you buat cantuman mata tunas pada penanti yang tak producing any floral stimulus to flowerings. Okay, this like macam terbalik sikit with the competency tadi, right? Competence tadi shoot, vegetative shoot, alright, you put on the stock plants that are ready to start flowerings. So the vegetative scion tadi dia akan start flowering. Alright, so terbalik sikit, if the pucuk tu, right, dah determines to flowerings and you do a graftings pada pokok stalk yang tak ready lagi untuk flowerings, dia akan still flowerings. See, tu yang dipanggil determine. The determination of the shoot tadi, dia nak flowering tu sangat tinggi sampai dia overpass the stalk yang memang belum ready lagi untuk start flowerings. Okay. Okay, for example, okay, pokok tobacco normally after 41 leaves ataupun 41 nodes, okay, dia akan start flowering. Okay, in an experiment to measure the floral determination of the axillary parts, flowering tobacco plants were decapitated just above the 34th leaf pada 34. Kita nak tahu sekarang pada pucuk ke berapa, pada daun ke berapa. Okay, daun ke 41 dia start flowering. Okay, tapi kita nak tahu daun berapakah, on what leaves, what number of leaves yang pucuk tu dah start determined to flowering. Kalau kita potong pun dia akan start still flowering. Alright, kita nak tengok daun ke berapa. So the first experiment was done on the daun ke 34. Okay. Daun 34 is, however, if the 34th bud was excised from the plants and either rooted or grafted onto a stalk without leaves near the base, it will produce nearly a competent set of leaves before flowerings. So means, okay, kalau buds tu not determined, okay, ni donor pokok yang daun, kalau you literally counting the leaves tu dia akan nombor 41 lah kat sini. 41. Alright. Sorry, 41. Right. So kalau you grafted pokok 1, 2, 3, 4, 5, 6, 7, 8. Pada tahun 34, Alright. So pada mata tunas kat sini 34, right? So dia akan still produce daun sampai ke 41 until 41st leaf, and then dia akan start flowering. If you do a rooted, okay, you tanam from apa ni? Vegetative stem macam tu. Okay, rooted. Okay, you letak hormon and dia keluar akar, dia akan grow sampai 41. Alright. Even you do a grafting here, dia akan start sampai 41. 41 then it start flowering. Means that pada daun 34, its results show that the 34th bud was not yet florally determined. So mana daun ke-34 tu dia tak ready lagi untuk flowering. So what happened? The second experiment, the first experiment, the second experiment, a donor plant was decapitated above the 37 leaf, atas daun ke-37. Alright. Okay, tadi A. And then this B. B ni kita nak tengok daun 37. Alright. Daun 37, donor, and then in situ, right? So maknanya daun 37 you masuk, dia akan still sampai ke 40, ni macam biasalah, dia akan sampai 41 kan flowering. Right? Right. Tapi kalau you start rooted ataupun grafted, alright, pada daun 34 tadi dia, kalau daun 34 dia akan grow macam biasa, rooted sampai 41, right? Kalau dah 30, kalau you try to take decapitated daun 37 ke atas, alright, and then you do a rooting here, rooting propagation, right? Dia sampai 41 dia akan flower. Right? Even you do a grafting, there's flower. So means that pokok tobacco ni, dia start determination, dia dah entering determination stage pada daun 37. So whatever you do with the daun pucuk with, daun ke-37 tu, donor tu, right? Donor tu, you ambil and then you buat grafting ke, you tanam rooting ke, dia akan terus flowering. Alright? Kalau you ambil daun 34 tu, dia tak akan start flowering lah. Okay? So, tu adalah determination. Dia sangat determined untuk flowering. Okay? Kalau dia tak determined, dia akan go back from the start sampai dia daun 41 dan baru dia akan start flowering. Okay, ni simple. I don't have to explain too much. Ni competent dan determined tadi. Alright. And the next, circadian clocks. Circadian clock ni kita dalam bahasa lain dia adalah biological clocks lah. Okay. Dia happens secara biologi. Macam mana nak cakap? Tanpa external factors. Okay. It's like macam kalau you pergi overseas, macam saya pernah pergi, saya duduk kat UK and then balik kat Malaysia, first few days tu memang berterabur sikit and then dah suit to the normal rhythm according to Malaysia times. Okay. And then bila saya went back to UK, my flight was pukul 1 pagi waktu Malaysia. Alright. Saya sampai kat UK pukul berapa? Oh, saya tak ingat lah. Katakanlah saya sampai kat UK pukul 7 pagi. Macam ni. Tapi saya tak ingat, tapi waktu pukul 6, saya dah start, 6 petang waktu UK, saya dah mengantuk dah. memang mengantuk sebab waktu tu kat Malaysia pukul 2 pagi. Saya kat Malaysia pukul 2 pagi memang dah tidur lah. So dia akan follow the biological clocks according to Malaysia. So bila pukul 6 petang tu saya dah start tidur, saya akan bangun pukul 2 pagi waktu UK, dan saya tak tahu nak buat apa-apa, tengok movie je lah pukul 2 pagi. So 2 pagi tu waktu Malaysia dah pukul 8. Pukul 8, pukul 9 pagi kan. 9 pagi kita bangun waktu Malaysia kan. So tapi kat UK masih 2 pagi. So saya memang terkejut bila pukul 2 pagi tu saya dah tak nak buat apa, tengok movie je lah, that's it. Right? So that is biological clocks. It will take a few days untuk go back to the normal condition, normal condition according to the current climate. Means the current climate that times, in my case was UK lah. Okay, kat UK. That's a biological clock ataupun circadian rhythm. Right. So rhythm behavior, leaf and petal movements, stomatal opening and closing, flower openings, growth and sporulation, pattern is fungi, daily changes in the rate of metabolic pathway. That is circadian processes. Means that waktu tidur, metabolic processes will be different to that of during the day. Right, during the day you need to do activities, perform active activities. The metabolic activities will be changed, right, compared to when you are sleeping. Okay, so for example, pokok Castanea corymbosa open during the day, go up during the night. That is circadian clocks. And then the fungi checks on the sporulation. Okay, the spot marks the few hours each day when the fungus produce spores. So that is the circadian clocks lah of the fungi ataupun sporulation. Dia ada contoh waktu siang dia akan do the sporulation after a few hours, and then during the nights, and then we stop doing the sporulation, and then dia akan start sporulation again after the normal times of doing the sporulations, come back again as for yesterday, right? As like yesterday, and then it will continue. That is circadian clocks. 3 parameters: amplitude, periods, and phase. Periods is between between the peaks lah. Okay, biasanya 24 hours. So kita periods ni depends on the species. Okay, so the peak tu different of the species lah. Tapi normally memang kita according to Earth punya times is 24 hours lah. Right? Ni okay, governed by internal pacemaker ataupun molecular clock mechanism called endogenous oscillators. Okay, bila dia ada oscillator ni dia akan play around with dia akan the endogenous oscillators tu is attributed to these patterns of the circadian rhythms. Alright. Kalau natural condition, oscillators strange to a true 24 hours by environmental signals. Kalau signal is removed, it's free running like that. Okay, so this one it's okay, don't have to go for more details. So circadian clock is like that. So it affects the, like I said, the set example says it determines the leaf and petal movement, determines the stomatal openings, closings, flower openings, growth and sporulation pattern in fungi, daily changes. Okay, these are some of the physiological behavior happens, all right, because of the circadian rhythms. That is the principle. Don't have to go more details, I guess. So phytochromes and cytochromes and train the clocks. Okay, so how circadian clocks triggered. One of it adalah bispecific wavelengths of light that can be induced. Phase shifting indicated that the light response must be mediated by specific photoreceptors. Okay, badan kita a flower ni dia tahu circadian clock dia, one of it adalah signal dia adalah matahari, right? So matahari mesti ada receptors on the leaves, a receptor in the leaves that can detects the changes of the light, okay, whether it's darkness ataupun during the day of having light, right? So kita panggil characterochromes CRY 1 and 2, alright. Okay, simple. And then photoperiods. Okay, if you realize that I actually go from one subtopic to another subtopics, and it's very on the right sequence. Okay, we learn on the what is the floral, what is the intro for the flowerings, and then we go for the flower organs, and then what genes that controls the setting of the flower organs, okay, ABC. And then we go for, okay, whether the plant has the competency or determination to start flowerings, and the factors that are controlling that, right? And then we go for the circadian clocks, that, part of it adalah a process that finally determines the flower is okay to start flowering or not, right? This circadian clocks, okay? And then we go for the photo periodisms, all right? This is when the terms short day plants and then long day terms, short term plants and long day plants are present. Periodisms. Photoperiodism is a physiological reaction of organisms to the lengths of day and night. Can be found in both animals and plants. In animal kingdoms, day lengths control such as seasonal activities as hibernation, developments of summer or winter coats, and reproductive activities. In plants include the initiation of the flowering, a sexual reproduction, formation of storage organs, and the onset of the dormancy. All right. This is one. There was one of the old experiments conducted in 1920. All right. Before that, numerous plant species flower during the long days of summer. Musim panas is logic. Okay, kita nampak pokok berbunga tu logik lah. Alright. Because of the first thought, probably is due to the accumulation of the photosynthetic products ataupun carbohydrate, right, synthesized during the long day. Long day, more light, more photosynthesis, so more carbohydrate. So it was thought that these carbohydrates and from the photosynthetic activities, high photosynthetic activities, all right, dia contributes to the start of flowerings. Okay, so bila conducted experiment in which the hypothesis was shown to be incorrect. Okay, and then in 2012, a mutant variety of tobacco, Maryland mammoth, grew to about 5 meters in height but failed to flower in the condition of summer. Okay. Okay. The small ones, if you see in the picture, is the wild types. Okay. Kalau yang sebelah kanan tu, mungkin yang mutant. Okay. Dia tumbuh tinggi, 5 meter high, tapi fail to flower in the condition of summer. However, plants flowered in the greenhouse during winter under natural light condition. Right, so bila dia buat letak when they put into in the greenhouse where the light is normally customized customized to 12 hours day, 12 hours night. So the plants flowerings. Okay. They concluded that day length was the factor in flowering rather than accumulation of photosynthetic products. So means that the day length means that the exposure of the lights on the towards the plants is actually contributes whether the plants can start flowering or not. Alright. Tu yang tu kita panggil photoperiodism. Okay, it's not because of the photosynthesis activities yang trigger the flowering stage, but it's actually how long the plants are exposed to the light. Two main categories, short day plants and long day plants. Short day plants flower only in short days of the flowering accelerated by short days. Long day plants flowers only in long days or their flowering is accelerated by long days. Okay, can skip. Can skip. Oh, can skip. Okay, this is the summary. This is the more, I think this is one of more explainable because got an image here. All right, so measuring the length of the night. Okay, here is short day plants. Okay, it's 20. Oh, sorry. Okay, it's 12 hours up and 12 hours down means that okay, bawah tu gelap, atas tu cerah. So the middle is 12 hours. So 12 hours each lah, right? So here is will be 0 will be 24 hours. Right? This one is 12 hours. Right? So if the short day plants is given, if a short day plants is given less darkness, okay, less than 12 hours of darkness, it will not flowering. It will not be flowering, right? If the short day plants is given the darkness more than 12 hours, all right, it will start flowering. All right. If the plants, right, if the short day plants is given more than 12 hours of darkness, like the previous plants, okay, it will flower. But if suddenly you give a flash of light during the darkness, dia takkan flower. You kacau sekejap pun dia takkan flower. Right? Even though the darkness hours ataupun the darkness periods is more than enough to initiate the flowering in the plants, but in case you interrupt the darkness with a flash of light, dia tak akan flower. Alright? They won't show their plants. Alright? So for long day plants, okay, again, it's 0, 12 hours is 24. Alright? So if you expose the plants if you expose the plants with the lights more than the plants under the darkness, it will start flowering. If you do the vice versa, the next more than the light exposure, it will not start flowering. Right? If you give, all right, darkness more than the light, right, it should not be flowering. Tapi if you give a night break, waktu gelap tu, even though you bagi 12 hours of darkness, sepatutnya dia tak flowering like this. Right? Sepatutnya tak flowering. Tapi katakanlah you bagi gelap pukul 12.30 malam sampai pukul 10 pagi. Alright? 12.30 malam sampai 12 tengah hari is 12 hours of darkness. Tapi kalau you bagi dalam pukul 3 hingga pukul 4, tak kisahlah, right? Pukul 3, 4 ke 4 ke 5 ke, you bagi light, you disrupted the rhythm with a light, right? Pokok akan flower. Alright, even though you expose more darkness than light, tapi you interrupted with a short period of light, pokok setiap akan flowering. Okay, dia contradict dengan short day plants. Cuma, it is important to note that night treatments of only a few minutes are effective in preventing flowering in many stages. Alright, so kat sini, see, for short day plants, it's enough just you give the plants 1 or 2 minutes, okay, few minutes is enough to break the prevent the flowerings in the short day plants. Tapi kalau pokok long day plants, you need a bit more longer lah, dalam 1 hour ke half an hour probably. Okay, required to promote flowering in LDP. Okay, long day plants. Okay, this is very important. This is a very important gambar aja, right? I repeat, this is very important that you have to understand whether the plants will be flowering or not, depending on the light exposure. Okay, see on the above part, is short day ataupun long night plants. Okay, kita panggil short day plants lah, right? The down here is long day plants. Okay, so we got what we got here: 0, 12 hours, 24 hours. All right. So start one by one. I repeat, this one is very important, right? To understand. Flower, the long day plants, you give more than 12 hours, right? They akan flowerings. The short day plants, of course, they receive a very less night punya darkness, very less night ataupun darkness exposure, dia tidak akan flower, right? Nevertheless, untuk yang second columns, the long day plants will not be flowering if you expose the plants, alright, less than the critical night length, okay, ataupun day length lah. Okay, sedikit dia lagi kurang daripada sini, dia tak akan flowering. But short day plants, dia akan flowering. Okay, so the short day and okay. RNI red light, FRNI far red light. Okay, red light will prevent the flowerings. Okay, red lights will prevent flowerings in the short day plants. Okay, far red light will induce ataupun trigger the flowerings in the short day plants. Okay, so here, even though you expose the night longer than the exposure of lights for the short day plants, but you interrupted with the red lights, it will not go flowering. All right. Tapi for the long day plants, alright, katakanlah lepas 12 jam, you turn off the light and will go for darkness, alright, and then suddenly you give the red lights, alright, pokok long day akan flower, alright. The fourth columns for the fourth bar, alright. Pokok long day plants, okay, you bagi the 12 hours, alright, 12 hours, and then you give the darkness, alright, you bagi red light, sepatutnya dia flower, alright, and then you bagi gelap balik, and suddenly you interrupted with far red light, so the long day takkan flower. Long day ni, the far red light dengan red light ni, far red light dengan red light ni dia bagi difference opposite effects on the short day plants and the long day plants. Okay, while the red light, okay, is favor for the long day plants to flowerings, tapi dia prevent short day plants from flowerings. Right, the far red light is nevertheless, okay, promotes flowerings in the short day plants, but will prevent the flowerings in the long day plants. Okay. So, sekejap. I want to make sure that I'm not confused. Okay, betul lah. So, go to the 1, 2, 3, 4, 5. The fifth bar. Alright, the final light. Okay, this picture. Alright, the timings always from down and moving up. Okay, so for this bar, okay, bila you start giving darkness and then you flash red light, okay, the long day plant dia akan berbunga, short day plants dia tak berbunga, and then suddenly you bagi balik after few times, and then you bagi far red light, right? The long day plant sepatutnya tak supposed to be not flower. Short day plant supposed to be flowerings, right? Because the far red lights favoring is in favor of short day plants to be flowerings. But if suddenly you give back the red lights, okay, dia akan promote baliklah the long day plants to flower, but will prevent the short day plants from flowerings. You have to always look at the final wavelengths ataupun light that is given to the plants. Okay, so the last is far red light. Is of course will promote the flowerings in the short day plants, but will prevent the flowerings in the long day plants. Okay, for this graph, for this image, faham tak?
Faham.
Ada. Faham Tak ada confusion No. Alright. So, ingat, far red lights and red lights for flowerings is different to that of for the germination requirements. Two difference. Okay, this one for flowerings. Okay, flowerings ni lah dia punya ni. Kalau germination lain, yang tukar receptor, same mechanisms, tapi effects of the far red lights and red lights for the germination dengan effect of flowerings on the short day and long day plants difference. Okay, dengan confuse. And then vernalization. Some plants need a specific temperature to start flowerings, right? Vernalization, kita effect of the we give we expose the plants in a low under a low temperature, right, to trigger them to start flowerings. Vernalization is a process whereby the depression of flowerings is elevated by cold treatments given to hydrated seeds of growing plants. All right. Vernalization process requires active metabolic processes. Without vernalization, plants that require this process will show delays in flowerings or remain vegetative. Okay. Characteristic of vernalization: plants differ considerably in the age of which they become sensitive to vernalization. Considerably. I feel complete. Oh.
Considerably.
Yeah. Considerably, right? Okay. Plant differ considerably in the age of which they become sensitive to vernalization. Dia sampai kena sampai stage dia sensitive to vernalization. Kalau dia tak sensitive, even you give the vernalization process, it will not respond. The effective temperature range for vernalization is from just below freezing to about 10. Just below freezing is freezing point is 0, right? Low temperatures between 0 to 10, right? So just below freezing, probably -4 to 10 lah. Sorry, -4 to 0 temperature. Vernalization effect can be lost due to exposure to high temperature, the vernalization process. So vernalization process is cold treatments, right? If you increase the temperature, of course, it's not a vernalization process anymore. It can be the vernalization process, right? Vernalization process result in competence to flower and pea genetics changes. Vernalization takes place primarily in blah blah blah. Okay, betul lah. Tu je nak bagi tahu, you kena ada treatments vernalization ataupun low temperature. Kalau in the growth rooms ataupun greenhouse, you need to induce them with the low temperature, alright, so that they can start flowering. If not, it will not flower. Arabidopsis got a genes as a repressor of flowerings, flowering locus-C, FLC. Okay, ni okay, you can read lah later. Dia pasal genes lah yang control macam flower tadi lah, four wheels organs tadi, right? So the principle is the same. So, right? At 20 degrees, for example, FLC expression. Dia itu pasal ekspresi. FLC expression. Okay. So before cold exposure, alright, FLC, the genes, alright, FLC ada repressor, means that it can repress the plants from flowerings. So during at 20 degrees before the cold exposure, the FLC expiration is high. All right. During the cold temperature exposure, all right, the FLC punya expression will go down. Will go down. So bila it go down, no more proteins that will inhibit the flowerings. All right. So so after you increase balik kepada 20 degree Celsius, dia akan start flowering. Tu je. So prolonged cold exposure you bagi cold exposure. So ni lebih kurang sama je lah. So I dah explain what is a mutants, how you do get a mutants, what's the function of studying the mutants, right? What happens if the plant is a mutant, you knock out ataupun you silence the genes that producing specific proteins for the specific function that you are looking, right? So that is same je lah. So ni yang gambar tadi kita dah discuss. Right, it's going down. Okay, autumn suhu masih dalam probably 15 to 20 degrees, and then during the winters can be low temperature to below freezing points. So vanillization happens here. Right, and then spring the temperature will go up again, and then dia akan start flowering. Alright, so during the summer slowly FLC expression dia akan meningkat balik, so the plants will not be flowering because of FLC genes produce the proteins that can repress the production of, I mean not the production of the flower, means that can prevent the plants from flowering. And then the temperature will start drops again, and then vanillization happens back here. Alright, and then during the springs the temperature will increase and keluar bunga balik. That's why spring kita panggil musim bunga. Right, musim bunga. Waktu autumn, alright, bunga akan start gugur, and then dia akan hibernate. During that times adalah vanillization processes. All the leaves will not be, depends on the plants. Kalau pokok daun yang pokok-pokok besar dan berdaun, dia akan gugur lah. Right, gugur. And then slowly after the winter akan masuk springs. Alright, the temperature will slowly increase, and then the flowers can start flowering again. Alright, so musim bunga adalah musim spring. So now you know why musim bunga kita panggil kenapa musim spring banyak bunga, because of there was cold vanillization during the winter, not few months before the spring, right? Simple. Simple. Okay. Grafting studies provided first evidence of transmissible floral stimulus. Okay. Florigen is the proteins. Okay, I think it's the proteins. Oh, it's a hormone. So hormon yang hormones yang terlibat dalam flowering process. Jadi simple. Because of the floral stimulus is transmitted along with sugar in the phloem, it is subject to source and cellulation. An induced leaf located close to shoot apex is more likely to cause flowering than an induced leaf at the base of the stems. Alright. Oh, I don't think you need this, but it's good for your information, for your further, um, for your knowledge. But yeah, for the examination, not really much I care. Alright. Okay, it's biochemical signal. Okay, good. I think that's all. Saya pun dah tak ada soalan. So kita dah tamat kita punya lecture, right? Exam soalan okey je, right? Cuma you have to understand look back on the photosynthesis and respiration. Okay, the principle of photosynthesis and respiration. Alright, make sure you know the basic concept of that. Right, the respiration and photosynthesis process. Right, and also you need to know, actually, if you realize, right, during my lecture, at some point in the lecture, I had repeated a few times, okay, certain not topics, but certain points, okay. That means I want you to really understand of that specific things that I was talking, and then that is because biasanya soalan macam tu, benda yang saya explain banyak kali tu dia akan masuk exam lah. Okay. Sebabnya bila dalam exam, saya akan masukkan benda-benda yang I think applied to you, understandable to you, and then you can bawa that principle, alright, even after you graduate. I mean, you akan faham the concept lah macam tu lah. So that's why ada few times saya akan repeat banyak kali, alright. Saya akan stresskan dia, alright. Ataupun mungkin saya explain it macam benda lah tu simple tapi it takes longer. Maknanya benda lah slide tu simple je tapi saya punya explanation tu sampai berjela-jela. Tu part of it juga, right? I'm not sure Dr. Latif, tapi that's my part lah, right? Films transport tu kena tengok juga, right? Film transport, subject topic film transport. Make sure you know the concept of film transport, right? And also important points I have just delivered in this lecture, right? Also also included in the final, right? Setuju je lah kot. Ada soalan? Kalau tak ada soalan, okey lah kot, kita boleh tutup lecture kita. Again, ikut track record, okay, memang student cakap memang pelang physio susah. It's not, susah bukan pasal soalan lah, tapi sebab subjek ni susah. Tapi sebenarnya fisiologi adalah basic lah, basic yang membuatkan you akan faham apa yang berlaku dalam plants, even basic not basic mechanism, it means the mechanisms dalam plants, even though you are not talking, you are not taking a direct subject related to plant physiology. Means that, for example, you go for, you are interested in natural product, for example, alright. The penghasilan of the products is a physiology. Dia ada external factors yang membuatkan plant tu defensif ataupun dia purposely menghasilkan that compounds, alright. So for what? So it's a plant physiology, right? You go for plants growth, then their relationship with the soil is a plant physiology. The different type of soil, right, and external factors, alright, the infestation, they can affect the physiology of the plants. If you are really interested in doing breedings, right, a plant physiology, right, you do molecular breedings, you introduce a genes, atau you do you study on the knockout genes, you study on the mutants. Of course, you want to know the you if you selected a genes, right, you do molecular, right. So you will study the genes punya mechanism. Bila sebut je mechanism, it will go back to the physiology, right. You study the nutrient study is a physiology. You cannot run the physiology from the physiology. It just, if you understand physiology, it will help you very much to increase your understanding, alright? Understandings, and then you boleh explain kepada orang lain sebab you faham the physiology mechanism, right? You tak apa orang kata, not just literally hafal fakta macam tu lah, right? So even you belajar anatomy, right? If for example, if you go for the FYP and you really like on doing on the anatomy, right? Anatomy ada dua, right? Mungkin dalam natural condition, you just check the characteristic of the plants, different plants, right? Looking at the characteristic, and then you want to do taxonomy, right? Then you do the anatomy, ataupun you study on the deep environmental factors on plants and looking at the changes of the anatomy, alright. So it's a plant physiology because of there is always a mechanism why. The question is always why. Why this the anatomical structure is different, right? What cause, what factors that contributes ataupun influence the the changes from the normal to a new modified anatomical structure is a physiology, right? The production of genes is still a physiology. How the genes work, still physiology, right? Means that you cannot run from the physiology. The fact that I am a plant physiologist, so I can easily explain to you the subject of the the subject of breeding, the subject of natural products, okay. Tak termasuk structure lah. I don't like study on the structure. So it means the natural product, how the product, how the compound is being produced, on what parts from the original compounds coming from what roots, right? What the roots compounds, okay? What the roots molecules. I can easily explain. I can easily explain about the nutrient management. What happens during the nutrient uptakes? Why nitrogen? Salah satu deficiency of nitrogen boleh ditengok pada daun tua, bukan daun muda, right? The deficiency of if you look at the plants, kalau daun warna kuning, definitely is not a nitrogen, because nitrogen is movable, right? Is that because of the ferrum? Normally ferrum is at besi. You will see the yellowish color on the young shoot. It's not because of the nitrogen, right? because of the pheromones. Because of what? Why pheromones? Because pheromones involve primarily in the production of the chlorophyll or involved in the photosynthesis process. It's a physiology. I can explain to you. I can explain to you. I can explain to other people outside with a very confidence, right? With very high confidence. So people looking at me and asking what this is, okay? In terms of kenapa pokok ni kuning, ke apa kan? I can explain very clearly and very confident because I do understand the physiology, alright? So I do understand, if not at a very detail, at least, you know, in the general, right? I'm not a plant pathologist macam Dr. Sabina who is working and focusing on the plant pathology penyakit, tapi I do understand the mechanism. If after the attack of the pathogen, what happens? I do understand. I can explain you molecular punya mechanism, because what? I do understand the physiology. Some people, they are working on the penyakit, dia tahu pasal penyakit je. Dia boleh cerita pasal physiology of the penyakit saja. Itu pun sometimes penyakit je lah dia cerita. Right? If you do understand physiology in general, you boleh hidup kat luar lah. Masuk jadi consultancy, jadi consultants, orang tanya pasal advice untuk kenapa pokok ni sekian-sekian, kenapa pokok sekian-sekian, you boleh jawab, right? Even you go masuk dalam research, you nak study on molecule, you boleh jawab, you boleh buat, right? Because of the physiology is mechanism. The question is not what. In the physiology, question is not what, is why. So be clear why, you will always looking the answer. Why this happen? Why this happen? Why this happen? That's a physiology. Physiology is, for me, it's not tough. Tapi itulah, dia ingat banyak sangat fact yang kena hafal. Tapi again, whatever subject, whatever field you are going to take after this, you cannot run from the physiology. Alright? Physiology sangat memudahkan you untuk memahami sesuatu benda sebab you faham the mechanism. If you do faham the mechanism, the rest is just, orang kata apa, di hujung jari. So kalau tak ada soalan, kita close close the class lah. Okay, to majlis. So I hope you do I wish all the best in your final exams. So don't forget to submit. Don't forget to submit any tasks that are given to you, right? It need to be submitted this week adalah hari yang last, okay? Tapi saya harap you takkan more than hari Khamis. Okay, Khamis will be the last. Jumaat, Sabtu, saya mungkin tak akan terima lagi new submission. Make sure you hantar sebelum tu sebab Jumaat dengan Sabtu saya kena analyze all the data and then distribute the CAM marks. Alright. Sesiapa yang hantar yang lambat lebih daripada tu will not be entertained lah. Right, for example, kalau lab membawa, I think dalam 20 marks. So, you have 5 labs, okay? Kalau you tak hantar satu labs, you will lose 5 marks. Okay, you akan dapat, you will lose 4 marks. Alright, so the maximum of the labs will be 16. And same goes to the assignments and presentations. Right, your midterms, the majority of you scored more than 10 out of 15, so it looks great, right? So bila you dah buat yang terbaik waktu midterms, I think more than 90% scored 10 hingga 15 markah. Alright, so don't jeopardize your chance of getting good marks by not submitting the assignments or labs, right? You dah buat yang terbaik tapi ada benda yang you terlepas pandang, dia agak sayang lah. Alright. Okay, then kalau tak ada kita yes. Uh-huh. Okay, so macam ni. Nombor satu, untuk nombor satu, germination dengan flowering different. The function, the effects of the red light, infrared light for germination dengan flowering berbeza. Itu nombor satu. Nombor dua, biasanya kita buat, kita panggil imbat, kita nak break the dormancy, kita letak dalam gelap. Kita bagi cold temperature. Biasanya kalau dalam biasa kita meletak peti ais, tapi yang ni kita bagi imbat dulu lah dalam gelap tu. Kita just to break the dormancy lah. Tapi kalau yang sepatutnya kita bagi dalam sejuk, right? Sejuk tu lagi okey. Tapi yang ni kita imbat, kita bagi dia dalam gelap lah, untuk break the dormancy. One of it, break the dormancy lah. Alright. Nombor tiga, if you realize, red light ke far red light yang akan induce, regardless of imbibe tak imbibe dalam 24 hours, cuma saya nak tanya, red light ke far red light yang bagi pencemahan yang tinggi? Far red light. Okay. One of the hypothesis one of the assumption yang saya boleh bagi adalah, for the germination, far red light ni, dia, kalau you look at the spectrum, far red light tu dia lagi mendekati infrared. Sebab tu far red, sebelah dia red light. If you look at the electromagnetic wavelength, you can Google it. I think I've shown you before. Electromagnetic wavelengths, dia tu frekuensi dia lagi rendah. Alright. And then mungkin suitable for the germination. Kalau far red light, dia punya energy a bit more tinggi, right? Mungkin tak sesuai untuk germination. Itu background reason lah. For example, dia kalau yang betul-betul physical reason adalah the changes of the phytochrome. So maknanya kalau you bagi far red light, dia akan changes to red light and then vice versa, right? Tapi the explanation behind that probably because of the energy, right? The far red lights favorable for the germination, red lights not favorable for the germination. Kalau you perasan kat situ, the method, you akan bagi red lights for 1 minute only. Kalau far red light, 5 minutes. Macam tu. Sebab far red light tu, the longer wavelengths, energy dia tu tak tinggi lah. So mungkin tak tinggi tu good for the okay for the to trigger the germination, right? Itu explanation yang saya huraikan lah. Tapi to answer your question, kenapa kita bagi yang waktu gelap, basically just to break the dormancy lah. Sebab tu kalau kat rumah, kalau ada biji benih, right, kalau you nak percepatkan dia punya ni, you letak dalam gelap. Ataupun nak lagi okey, you masukkan dalam peti ais, tutup gelap, dalam masa 1 hari lebih, and then you keluar, and then you letak kawasan yang biasa lah. And then insyaAllah dia okey lah. Tapi depends on the seed jugalah. Kadang seed senang je nak pecah and then keluar, walaupun dia tak ada imbibition, ataupun kita letak in darkness, ataupun kita letak dalam fridge, dia senang je keluar. Ada seed yang macam yang keras, macam seed chili ke, kan, seed Arabidopsis ke, it needs to be treated with the dark ataupun cold temperature untuk break the dormancy. Kita bagi apa? Stratification. Saya tak ingat. Kita bagi cold temperature. Okay. No, white light actually ada 7 spektrum, alright? White light ada 7 spektrum. So, dalam 7 spektrum tu dia ada far red light, ada red light. Okay, ada spektrum kalau you look at the wavelengths, visible wavelengths, cahaya putih sekarang ni dia ada 7 colors, alright? 7 colors tu because of the different lengths of the wavelengths, alright? Sebab tu dia menghasilkan 7 color yang berbeza. So the highest energy adalah blue light, alright? The blue light, and then the lowest energy adalah the far red light. And then apa? I show you the menggunakan explanation sekejap ya. Okay, so so kita punya visible light kat sini. So kita punya yang biru tu the highest energy within the visible wavelengths. Okay, if you look at the short wavelengths contain more energy. Okay, on the far left side tu adalah gamma rays is the shortest one. Maknanya contains the highest energy, means that the energy yang gamma rays ni ada, dia boleh penetrate. I tak ingat lah berapa tebal punya konkrit. Alright, X-ray boleh tembus kita punya paru-paru, kita punya kulit, isi otot tak termasuk tulang, sebab tu dia boleh nampak tulang. Alright, and then UV ni boleh masuk lapisan nipis pada kita punya kulit, tapi dia boleh masuk sel jugalah kan, sel kulit tu. Alright, visible wavelengths cannot penetrate into our skin. Alright, infrared lagi lah tak boleh masuk penetrate into our skin. So visible wavelengths ni adalah white light. Cuma Tuhan tak jadikanlah white light ni terus 7 color. Cuba bayangkan kalau Tuhan jadikan 7 color, berpindah-pindah mata kita tengok semua bercolor-color. So that's why Tuhan bagi luxina warna putih je lah. So actually dalam warna putih tu ada 7 spectrums, which is blue light and far red light, the farthest. Okay, so kalau wavelengths blue tu disukai oleh chlorophyll, akan diabsorb oleh chlorophyll. That's why leaves you tak nampak warna blue, because dia dah diabsorb oleh chlorophyll. Alright, so green dia akan reflect. Okay, yang merah itulah yang digunakan untuk merah dengan far red tu yang akan play important role in the flowering process yang kita belajar tadi, short day and long day plants, and also triggered for the germination. Alright, macam tu. Faham? Dia sama je, kita bagi far red light ke dia amount dia tak jauh beza pun. Dia macam kita bagi far red light, kita tak increase pun dia punya intensity. The intensity of far red light dalam white light dengan light yang far red light kita bagi tu, amount dia lebih kurang sama je lah. Cuma white light tu mungkin rendah sikit ataupun tak rendah sikit sebab ada blue light tercampur kan. Itulah. Okay, so yang all the lab report tu simple je. Saya dah bagi instruction, jangan terlalu memeningkan kepala saya dengan banyak muka surat. Alright, and then 1.5 spacing macam tu lah. Okay, so buat saya tak expect too much details, too much words, as long as you punya pengisian tu dapat memberitahu saya dan you faham. Alright, Walaupun tak 100% saya boleh expect lah, because we didn't have the face to face punya lab, so I do really understand lah. Alright, tapi at least you show me, okay, you understand what is this, what is that, and then itu okay, walaupun discussion tu tak adalah sampai satu muka surat, no, itu terlalu banyak dah bagi saya. Alright, ada soalan lagi? Alright, kalau tak faham for any subject, walaupun subjek tu Dr. Latif, sebab kita share subjek, tapi previously subjek tu memang saya yang ambil lah, saya yang mengajar the whole. Alright, tapi kali ni Dr. Latif dengan Dr. Hairul bantu saya. Right, so if you don't understand any topics in the 14 weeks punya subject, you just boleh contact, boleh bagi tahu dalam groups, ataupun boleh you boleh contact saya directly lah. So tak ada masalah. Yeah, physiology is my forte lah. Okay, kalau tak ada apa, kita stop lah. Jumpa lagi tak tahu bila. Di masa akan datang, insyaAllah face to face lah kot. Itu kalau I share the subject you are taking in the future.